Okay, today we're going to be talking about organic compounds. This is Lecture 2, Part 2, and it will complete Chapter 2 in your book. So, hydrogen and other elements are covalently bonded. This means that they are sharing that outer valence electron or more than one of those electrons. So, completing that outer valence, making the molecule stable. So what are we talking about? We're talking about carbohydrates, which you know are also starches and sugars. Lipids are fats. Proteins are things that are have amino acids that are the building blocks. And then we also have nucleic acids, which are the beginning pieces of DNA, like adenine, thymine, guanine, and cytosine. So we have some reactions that have to take place in order for these particular molecules to be formed. So we want to form polymers from subunits. Those subunits are referred to as monomers. When we're talking about dehydration synthesis, it's a condensation reaction. So we have the removal of water. And I always remember that because when you're dehydrated, you lose water. Synthesis, as hopefully you recall, is the making of. That's what that means. When you form polymers from subunits of monomers, you have the removal of water, or the OH from one molecule and an H from another. It does require some energy. Energy is always in the form of, let's write that out, ATP. Not very neatly. So if we take glucose here, and we add it with fructose, we get sucrose, which is table sugar. We get rid of a water, as we talked about. And you'll see as I jumped ahead that formula, the chemical equation there is at the bottom. Hydrolysis is where you add water to break polymers into their smaller units. Okay, so the water is what is used to break those bonds. So when you may have studied this Gibbs free energy when you took chemistry, when you take a large molecule like glucose or lactose, as we have here, you add water, those hydrogen or not necessarily hydrogen, but the bonds are broken, and that's what the water's role is. It releases energy. So if you think about that, when you have glucose and you're executing cellular respiration, you want to break those bonds because that's where that energy is, and that kind of ties back into the whole potential energy in your orbitals. So here we have lactose, and we're adding water, and our end results are glucose and galactose. So carbohydrates, what are they? They are the main source of energy for most living things. That helps us to understand our relationship with plants. We have compounds of carbon, hydrogen, and oxygen. So our most common one that you're familiar with, and you've seen me write this formula before, is C6H12O6. Okay, so we have our monosaccharides. Those are simple sugars like glucose, fructose. And there's the chemical formulas. Ribose and deoxyribose are pentoses, so they're a 5-carbon sugar. Carbohydrates are anything from pasta to bread to crackers to potatoes. They're a disaccharide, which has 2 monosaccharides combined. So here you can see we take lactose and glucose, and we get galactose, which is the common milk sugar. If we take maltose and break it down, we get glucose and glucose. Starch hydrolysis product. So now you have sucrose, and that's broken down into glucose and fructose. Sucrose is a table sugar. Now, we will talk about here shortly enzymes and their role. Sugars always end, and I'm sure you've been taught this in OSE. That's a key if you see a word you're unsure of, and you see that OSE, most likely is a carbohydrate or a sugar. Now, we know that enzymes are specific and that there are only certain ones that certain enzymes that break down certain substrates. So maltase is going to break down maltose because it's specific. And what helps us to remember that whole lock in key theory. Okay. Carbohydrates can also be in the form of a fiber. Polysaccharides are many-chained monosaccharides. Starch is a great example of a polysaccharide. That's how plants store the energy that they're making. So a lot of times kids get mixed up and they think that potato is the plant, the potato is the root, it grows underground, and it's that plant that's growing above ground, executing photosynthesis, making that glucose and shipping it down to the roots, and it gets stored. That's your potato. Glycogen is in your muscles. That is a short-term energy storage in animals. Cellulose. Now, cellulose is unique a component of plants. The way to remember this is it's that undigestible carbohydrate or that piece. And the way it's like in the corn hulls, around the corn, you cannot digest those. Or even celery. It's that stringy part when you bite celery. That's where your cellulose is. Unlike glucose, linkages in starch and glycogen and those in cellulose cannot be hydrolyzed by most animals. Hydrolyzed means they cannot be broken down. Lipids are, in fact, triglycerides, oils, waxes, and steroids. They are a class of compounds that are insoluble. We talked about this in the last lecture. They're hydrophobic, so they're afraid of water. And you could do an exercise where you put some fat and water and you try to shake it up. You know that those lipid molecules separate out. What do they function to do? They're a great source of energy, providing 9 calories per gram. That's how much energy they would provide the body, as opposed to carbohydrates, which only provide 4, and we'll get into detail on that. In structure, they are a major part of the cell membrane, where the phospholipids. So you know we have that bilipid layer, and you have those phospholipids that are all lined up. So you have 2 of these, one here, and then you have this one that goes that way. Oops, no, I did that wrong. You know what? Oh, I'm not going to erase that, so I'm going to cross that out. And it's actually like that. I did that incorrectly. And this area is your water loving. And this area here is your water hating. Okay? Okay. Right, moving on with lipids, and here's what I was talking about. At the top part, lipids, the main components of a lipid are 3 fatty acids plus a glycerol. So you have your glycerol is at the top, and then you have your fatty acid strands which hang down from it. So fatty acids are those long straight chains of hydrocarbons. They have a terminal carboxyl end. Your saturated fatty acids have no double bonds. They pack easily and are solids at room temperature. So that's like butter. Monosaturated, has 1 double bond. Polyunsaturated has 1 greater than 1 double bond. Double bond causes a kink. So that kink, as opposed to where you're saturated, which are your fatty acids, which can pack right up tightly. Here, your butter. Okay. This polyunsaturated, they don't pack up. So that's your liquid. That is oil. And these are common in vegetables and fish. Trans fats are partially hydrogenated vegetable oils. They actually increase the risk of CHD, which is coronary heart disease. You find these in cakes, cookies, margarine. So what happens is you are chemically changing the oil to become solid a vegetable oil like Crisco or margarine, right here, to become a solid at room temperature, because you're inserting hydrogen bonds, straightening out those kinks, so to speak. Triglyceride is your neutral fat. It's fat that you find on your food label. It is your reservoir of energy. As I already stated, lipids are 3 fatty acids and a glycerol. They're stored in your adipose tissue and muscle. When you burn it, I already explained this, you get twice the yield because it has 9 calories as opposed to a carbohydrate which has 4. And here's what I was talking about before with your bilipid layer. Here is a great section of a cell membrane. Here are your hydrophilic edges, and here are your hydrophilic, and in here is your hydrophobic. Okay? So phospholipids are an important part of the membrane. You have 2 fatty acids and a glycerol here, and a phosphate. So unlike your lipid that has 3 fatty acids plus a glycerol, in a phospholipid, you only have the 2 fatty acids plus a glycerol, and you have your phosphate. What else are lipids for? Well, we also see them in the production of sterols in their derivatives. Sterols background is a lipid. There's no fatty acid. It's a 4-ring structure. Sterols are how we make up our cholesterol. There's 2 sources. One comes from your diet, and the other is genetic. There's some people that have very high cholesterol rates but are exceptionally healthy, exercise, eat a proper diet, but yet their cholesterol is off the charts, and this is primarily caused by genetics. So high blood cholesterol ends up leading to atherosclerosis, and that is where you have the plaque buildup in your arteries, clogging your arteries, decreasing the circumference of that vessel so that the heart has to work harder to push the blood through to the rest of the parts of your body. You have different types of lipoproteins. LDLs are low density. This is your bad cholesterol. It can be, it's also can be modified to form your sex hormones. So those sterols also are involved in the production of not only cholesterol but also estrogen and testosterone. Proteins. Proteins have the greatest amount of diversity. Why? Well, because you know your amino acids are the building blocks of proteins, okay? So it is the arrangement of those amino acids right here that determine the function of that protein. And we're going to talk more about that. I already mentioned carboxyl groups, and I just want to make sure that you recall that from chemistry. Your carboxyl group is COOH. Your amine group always has that nitrogen end, and that is your NH2. The components are carbon, hydrogen, and oxygen, building blocks of amino acids, as I said. Proteins are joined together by peptide bonds, which are indicated here in the diagram. How are they formed? Again, by dehydration, synthesis, and the covalent bonding. So a dipeptide, just like a disaccharide, is 2 amino acids that are linked. A polypeptide, like a polysaccharide, is 3 or more amino acids that are linked. Proteins are considered large polymers of 1 or more polypeptide chain. Now we're going to talk about the different structures of proteins. In your book, you will note that there is quite a bit of good information. I'm going to reference page 40 in your book. This particular page has a lot of information on the chemical structures for the 20 basic building blocks, 20 basic amino acid building blocks. So a polypeptide chain looks like this. You have your your terminus, I'm getting ahead of myself, N terminus and your C terminus. Okay, your N terminus makes sense. That's where your nitrogen is. Your C terminus has to do with that carboxyl group. Remember, it's peptide bonded backbones. The amino acids are joined together by those peptide bonds, and their arrangement will form the different types of amino acids. And you may remember this from a living environment: glycine, alanine, serine. And if you recall, you use that the wheel and you had to find the codons, like let's see, let's do A, G, C, okay, and that was a codon, and you had to use the codon to figure out, and it was, I think it was 1, 2, 3, yeah, so you had to use that to figure out what the amino acid was, and then you chain them together. And as we move through our units, we'll begin to talk more about that as well. So what are the functions of proteins? Well, structurally, it is keratin and collagen, like in your hair and your nails. It is for transport. Hemoglobin has a huge role. Hemoglobin has to do with your red blood cells. Yep, if I could, what if we put a B in there first, huh? RBCs are red blood cells. The hemoglobin is responsible for carrying the oxygen. It also has to have that high iron content there to make that work well. If you have low iron or low hemoglobin, you are considered to be anemic. That is a condition. Albumin is also important for transportation. Proteins also are regulatory in your hormones and your insulin. So, like insulin, I should say. So, insulin is a hormone. It is protein based. It's made in your pancreas. What is its job? It regulates. It regulates. Yeah, let's get our pen working down here. Blood sugar. And I'm sure you've learned that if you are have diabetes type 1, you have no insulin that is produced in the pancreas. Diabetes type 2, you have insulin that's produced, but it's misshapen. So it has difficulty in taking up the blood sugar out of the blood. Protective, this is also, proteins also function as antibodies, and these are made by white blood cells. Okay, and really the thing to note here, an important piece about it, about proteins, is that they're very specific. So their arrangement is specific to their function, and that specificity carries through in what they do in the body. We know that insulin has to be very specific. There are certain target cells. On the outside of the cell membranes that accept those hormones. The antibody-antigen relationship is very specific as well. We've also talked a little bit about enzymes, and we will talk more about that on this slide. So enzymes are also proteins that speed up chemical reactions. They're also known as a biological catalyst. They work on a substrate to either break large particles down, or make smaller particles into larger particles. Enzymes facilitate chemical reactions, and here is, right down here in this lovely diagram, okay, is that whole lock and key idea, where you have your enzyme, you have two small particles, they're joined together to form a product or a larger particle. So the product is the end result of an enzyme acting on its substrate. Amylase acts on starch to produce maltose. The active site is the spot on the enzyme that binds it to the substrate. Enzymes, as I said, speed the rate at which reactions occur. So you have reactants, okay, so here's your reactants, you begin. The activation energy without an enzyme is far greater than with an enzyme, okay, so it requires more energy if you don't have the enzyme than it does if you have the enzyme. Now, there are things that, so basically what am I saying? I'm saying that enzymes lower the activation and energy. Enzymes are not altered. They don't change. You don't have less of them because they've gone through a reaction. They remain the same. What influences that enzyme activity? There are certain things: temperature, pH, and concentration. And this is something that I'm sure you learned in Living Environment. temperature, the optimal temperature for enzyme reaction rates is 37 degrees C. That is your body temperature. You commonly may refer to it as 98.6 degrees C, or 2 degrees Fahrenheit. If the temperature is altered, you'll see here is the rate of the reaction is moving at a steady state as we reach 37 degrees C. And here is the optimal. As you move over that, the rate of reaction begins to decrease. Why? Because the enzymes become denatured. The same applies to pH. Okay, you see that it works at a good rate, gets to an optimal pH of about 7. Okay, and then it begins to decrease. Now this doesn't, there are exceptions for pH especially, like in the stomach acid acts on the enzyme pepsin. Pepsin requires a pH of about 2. Okay, concentration also. If you have too much substrate, not enough enzyme, that enzyme cannot work quickly enough because it's saturated. Another one would be your cofactors like magnesium. So now let's talk about the structure of proteins. There's primary, secondary. Oh, I knew that was going to happen. Hang on. Let's go back a slide. There we go. Okay, primary structure is the sequence of amino acids. It is unique. We've talked about that. The way that amino acids are arranged determine its function. The secondary piece of the structure are that the hydrogen bonds form between the different parts of the polypeptide chain, allowing it to fold. So going back to the primary structure of the protein, it has to do strictly with the amino acid sequence, okay? And we utilize that codon that I talked about, and you code for certain amino acids. The secondary structure, it describes how the chain of amino acids is actually oriented in its space. A common secondary structure of proteins is referred to as an alpha helix. The helix is what is that right-hand spiral destabilized by the hydrogen bonds between the amino acids at regular intervals. A flat ribbon is called the beta sheet. Okay, that beta sheet is formed when the hydrogen bonds join two primary sequences side by side. Okay, so I have that, both of those there, the alpha helix and the beta helix. All right, so now let's continue on with the tertiary. The tertiary structure is actually coiled and twisted polypeptide. And how you refer to this is like it's a globulin. A protein twists and folds to form three-dimensional shape. And again, as I'm telling you, it's more like a globule shape. Folding is a result of the interactions between the R groups. And then you have finally your quaternary. And this refers to how many polypeptide chains make up the protein, if there is more than one, and how they associate with each other. Proteins can become denatured. Denatured means that they have been altered. They lose their shape. If they lose their shape, they lose their function. And why do we know that? Because it goes back to that whole lock and key system theory, and which tells us that one, they're very specific. It has to fit together, just like a puzzle piece. All right, moving on to nucleic acids. Nucleic acids has to do with DNA and RNA. We have base pairing in DNA. That adenine goes with thymine, guanine goes with cytosine. Nucleotides are the building blocks for nucleic acids. When we're looking at DNA, we know there's a pentose, a phosphate, and that nitrogenous base, and that makes up that like ladder formation that you've heard teachers refer to in the past. Uracil, remember, is in RNA only. So here's that ladder formation. It's double-stranded for DNA. You know it contains the sugar deoxyribose. The sequence is unique to each species. Hydrogen bonds join the strands together at those nucleic acids. And again, remember that it contains thymine, not uracil. RNA is usually single-stranded, and this has all kinds of RNA have that ribose sugar. RNAs are a crucial role in which we are transcribing and translating our genetic information to build our proteins. Recall here again that it does contain uracil, not thymine. ATP is our energy. It is what we have to have in order to maintain homeostasis on one degree or another. Here I'm telling you it's your currency for cellular energy. Why? Because it is what cellular respiration generates for us to function on a daily basis. So here we have our adenosine triphosphate. Here's our chemical structure. It is filled with high energy bonds. When the third phosphate group of the ATP is removed by hydrolysis, energy is released. So the ATP then goes through phosphorylation. Phosphorylation, hard word to say. So when you remove that phosphate group, you change the ATP to the ADP. Hey, we are now at chapter 3. Excellent. So this concludes part 2 of lecture 2. I hope you found this invigorating, all of this information. If you have any questions, as always, please know that you can personal message me, email me, utilize the Ask the Class section to help you in your endeavors with this information. Thanks a lot. Have a good day.